Okay, so continuing on, we are approaching an equilibrium, right? You can see there's no areas there that are real heavily concentrated in that red dye. So I think we're done with this ridiculously. I don't know. Hopefully that helps you. Just something to grab onto, some phenomena that you can see, and when you reach what we call equilibrium, right? It's hopefully that's some demonstration of diffusion that you can kind of relate to. Equilibrium is when, again, there's no gradient anymore. There's no net diffusion. So there's as much moving through one side of the cell membrane as there is moving back the other way. Okay, now, so as we've talked a little bit about this, some substances can naturally diffuse through the plasma membrane. Others need some help, what we call facilitated transport. So facilitated transport is where we have some other molecule that allows the diffusion to occur through the membrane. A lot of times we'll have some sort of a protein, and I don't think we have to get too in depth here about how these work and what causes them to work. I'm comfortable talking about that there's a channel protein, or the classification of what we call channel proteins, that there's basically kind of these serve as a gate, if you will, where there's this channel of water or highly polar substance that allows molecules to move through the protein from the extracellular space to inside of the cell, into the cytoplasm. So kind of, again, just a direct channel or gate into the cell, specific for certain types of molecules or certain nutrients that you might need. Sometimes it's what we call a carrier protein that basically will fuse to a certain nutrient or ion or molecule that is needed within the cell, but that particular molecule can't get into the cell through the plasma membrane. Even though there is a gradient, there's a higher concentration outside the cell than there is inside the cell. So it will naturally move into the cell, but it can't get through the plasma membrane. So one of these, some sort of, typically a protein, will allow that facilitated transport. A carrier protein will fuse to the molecule and allow it to then slide through the polar, non-polar interactions of the membrane. Okay. Now, osmosis, this is diffusion, but specifically osmosis, we're referring to the diffusion of water. Water will move to, don't think about it as much as higher concentrations of water. Think about it as water will move to, higher concentrations of dissolved molecules. The water wants to get in there and dissolve those molecules more, if you will. So the water will spontaneously diffuse through osmosis to a higher concentrated area. And we have this term we call tonicity, I guess is the proper way to say that. Tonicity describes how an extracellular solution can change a cell's volume by affecting the osmosis. So tonicity refers to whether that, if I put a cell in a substance, it refers to whether that the cell will take on water or will lose water to the substance. So, and this diagram is from your text, talking about if we put red blood cells in a hypertonic solution, and true of just about any cells, but we can only see it with red blood cells because they're kind of like these red balloons. If I put it in a hypertonic solution, it's going to lose water and they're going to shrivel up. If we put it in a hypotonic solution, those cells are going to kind of blow up, and some of them might even burst because they're going to take on more and more water. In the next video, we'll talk about why that's the case. So, next video.